This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Hi everybody, good afternoon. And today we're going to continue talking about variant and mutation analysis. Just before we get to it, just a short announcement. Our server is running out of memory again, so we'll go for another reboot today at 5 p.m. Just make sure you save all your work there so you don't lose anything, okay? So 5 p.m. today, we're going for a reboot. Please back up everything you have there. And just to have a quick recap, so this week we started talking about variant analysis, variant detection. So on Wednesday, we talked about, okay, given a experimental and computational approach for variant detection, we talked about SNP chips or SNP arrays, which are an experimental approach to detect known variants. And also we talked about the computationally, how do we go from resequencing to actually identifying variants from probabilistic genotyping, which is a process called variant calling. And today we're going to talk about how do we answer the questions, how to predict effects of missense mutations in proteins. And for that, we're going to talk about computation approaches, both sequence-based and structure-based, and those that are qualitative, trying to ask the question, how do we identify pathogenic missense variants, and quantitative, how much a given mutation is affecting, for instance, a protein stability, protein interactions. And we also cover a few important mutation databases. So we also chatted about why are we studying mutations. And the main reason is mutations can give rise to different phenotypes. Some of them are very, very important for human health. So for instance, mutations are related to cancer risk and cancer predisposition. A lot of mutations are also related to inherited diseases. And this is a major concern these days. Mutations are also part of the process of emergence of drug resistance in pathogens and also in cancer. So the current situation is we take a long time to develop new drugs. However, pathogens are always evolving and trying to survive. Drug resistance is emerging very, very quickly, so we need new computational methods to inform better drug development. And understanding mutation effects is the first step towards developing better drugs, but also understanding disease and disease risk and developing better and more personalized treatments, as well understanding pathogenesis mechanisms. So we're going to talk a little bit about these today. So we need to understand the effects of mutations in order to move from treating the disease to in order to treat the actual patient and consider their mutations and their genetic background. And for that, we need to develop new tools, okay? And we also mentioned that mutations have different biological effects, and we were interested in a specific type of mutation, so small changes called single nucleotide polymorphisms, or SNPs. We are just one change of a nucleotide in the genome. And depending on where it occurs, we mentioned that this could be either on a protein coding region, which could potentially affect protein structure and function, and on non-coding regions, it could affect regulation. And when we do have a polymorphism on a protein coding region, we might ask the question, is this mutation pathogenic? Or we can ask the question, okay, it's pathogenic, but by which molecular mechanism? Is this affecting protein stability? It's affecting interactions between the protein and its partners. And this particular type of mutation on protein coding regions are important because not only proteins orchestrate molecular the molecular machinery in the cell, but also are important drug targets. So understanding how mutations affect proteins, protein structures, and protein functions are extremely important. And we also characterized these SNPs in different classes. So we have synonymous and non-synonymous SNPs. Synonymous SNPs are those SNPs in codons where you change the codon, you have still the same residue, and you have non-synonymous SNPs that it could be a change to a stop codon, so you have protein truncation, that's called a nonsense mutation, or you can have missense mutations, which is a change from one amino acid residue to the other, which are very, very interesting because they can be very subtle effects, which might also be related to pathogenesis, but also because they are very, very difficult to predict. So we'll focus on them today, missense variations. So, given a set of mutations, the first thing we can ask, for instance, we did the variant calling, or we did a SNP chip or SNP array, we have a sample with a set of missense mutations. We can ask the questions, are these mutations known? And what we can do is look up to specific mutation databases. So for instance, we can search, is this mutation associated with a specific phenotype, which could be, for instance, a disease association. Okay? And we can ask the question, is this mutation common in cancer? Does it play a role in tumorigenesis? We can ask the questions, for instance, is this mutation rare or frequent in a given population? So, for instance, considering the human population or specific subpopulations, American admixed, European, is this frequent in a given population? It's different from one population to the other. And asking the question, what mutation frequency in these populations can tell us, for instance, from a drug development perspective. And for that, we can look up different types of mutation databases. And we can basically categorize those into 4 classes. We have a lot of databases on human inherited diseases, which are mostly focused on germline mutations, so those that we are born with, and they are inherited from generation to generation. And we're going to mention one of them here. There are also cancer mutation databases. And of course, they are mostly focused on somatic mutations, which are those that we accumulate over life, which are very important for cancer. We can have also population variation databases, and also databases that try to characterize the effects of the mutation on the protein level. For instance, what's the mutation doing to the protein? Is it affecting its thermodynamics, its stability? Is it affecting its interaction with its partners? And usually we have data sets of experimentally characterized mutations. And as you can imagine, it's a very, very laborious and costly process. But it's a very important data. For instance, data on protein stability effects and protein-protein interactions, for instance. Okay? So one of the first mutation databases I'd like to discuss is the Human Gene Mutation Database, which is a very comprehensive resource on data on human inherited diseases associated mutations. And it's a data set collected from the literature every 6 months or 3 months, and mostly focused on germline mutations. And to this date, it has over 100,000 annotated missense mutations. So, for instance, if someone is interested in developing a new method to understand a specific inherited mutation, let's say you're interested in a metabolic disease, you can go to OMIN and select a subset of mutations that are common or they are associated with that specific disease. So the Human Gene Mutation Database offers a range of different phenotypes. The 3 most important classes there, I think, is metabolic diseases. We're going to talk about one of them today, developmental and also nervous system disorders. Another very important mutation database is COSMIC, is the catalog of somatic mutations in cancer. So COSMIC, as the name says, is focused on somatic mutations and is a very, very comprehensive resource with over 1 million tumor samples there. So either whole genome or exome sequencing from tumor samples from different types of tumors, and that accounts to this date for over 6 million coding mutations. So what happens in cancer, usually you have a starter mutation, what's called a driver mutation that actually started the whole process. And during this controlled replication, the tumor starts accumulating other mutations which are called passengers. So between these 6 million, you have a mixture of driver mutations, which are extremely relevant and important for diagnostics, for understanding cancer risk, as well as passenger mutations. And the main challenge is how you can differentiate between a passenger mutation and a driver mutation. And this is for multiple cancer types. For instance, you can ask the question, does my mutation appear in breast cancer or in lung cancer, for instance? This is also literature-derived information maintained by the Sanger Institutes. And the 3rd class of databases are population variation databases, and the main one and the most comprehensive one is NOMAD. So NOMAD is a genome aggregation database that originally was derived from ESAC, which is a hexome aggregation consortium. And NOMAD then has data on exomes, so the coding regions, and whole genome data as well, accounting for over 140,000 individuals. So it's a really, really large-scale data set. And this accounts for healthy population variation, okay? From different ancestries. While ESAC, the previous database was mostly focused on European ancestry, NOMAD brings other ancestries as well, including Latino, which is mixed, African, and so on and so forth. So these allow us, for instance, to ask the question, are there variants that are predominant in a specific population? And for instance, when you're developing a specific type of drug, how these variants affect drug efficiency. So this is very useful for a reference set for allele frequency, and it's maintained by the Broad Institute, so open source data. And the final class of mutation database are those experimentally measured effects on protein level. And these are extremely important, and all of these databases are important when you're trying to develop new methods. For instance, if you can use machine learning methods to use these as evidence to train novel methods. And we'll discuss this on week 12. So there's an important set of experimentally characterized data sets which are linked to protein structures. Why this is quite important? Because proteins are macromolecules that fold into a very specific tertiary structure, 3D structure. And this 3D structure is extremely important for the protein to perform its function. So you can imagine that mutations affecting proteins can affect the structure in different ways. And that's the purpose of these different databases, characterizing what specific way a mutation is affecting a protein function. For instance, we have different molecular mechanisms, and all of these data sets are quantitative. Is how much a specific missense mutation is affecting, for instance, protein-protein interaction. And in this case here, we have a protein complex. Each color here is a different protein that come together in a big complex, in this case, 6 different proteins, to exert a given function. So if you have a mutation that prohibits this yellow one here to come together, then you could have a pathogenic mutation, for instance. So one of the first databases linking mutations and changes, for instance, in protein-protein interactions. So whether a mutation that it's increasing or decreasing the affinity between the mutation in its other protein partners was SCAMPI database. And for each of those mutations, they have experimentally characterized how much is the interaction is perturbed and all the structural information as well. So more than just having the sequence, you also have the structure. And sequence structures tells different stories. And there are different molecular mechanisms then the mutations can affect protein structure. One of them is protein-protein interactions. The other one is protein-ligand interactions. And the first and the only structural database currently available is called Platinum Database. It is a very important database because I developed it. No, I'm just kidding. That's not the reason, but it's one important database because it deals with protein-ligand interactions, small molecules. And that has to do with protein-drug interactions as well. So if you're developing a new machine learning method, you need experimental data to learn from. And if you want to predict, for instance, whether a mutation is causing drug resistance, this is the kind of information you need. You need to understand experimentally how mutations are affecting protein-ligand interactions. And that's very important, for instance, for predicting whether a given strain of a bacteria, for instance, is resistant, but also considering that between each other we have different proteins. How would we respond to treatment? Okay? So whether a drug will be more or less efficient for each of us. And there are also mutation databases aiming for protein stability and interactions between protein nucleic acids, which are proterm and pronits. And they're really, really important resources. And based on all these databases, a lot of computational methods were developed. So the accumulation of this biological data actually allowed for the development of computational methods. And so considering that let's say we have a completely novel mutation. So if we have a known mutation, what we can do is search for it in different databases and see what are the known associations. But if we have a novel one, what we could do is use computational methods to actually predict their effects. And the computational approaches can then be divided into two groups: the qualitative ones, which is basically those that answer the question, is this mutation pathogenic or not? And the quantitative ones, which are asking the question, how much is this mutation affecting, for instance, protein stability or protein affinity? Okay, so more than saying this is pathogenic, so this is going to increase the risk of developing cancer, how is it doing in a molecular level. And furthermore, the computational approaches can be divided into two groups: those that only look at sequence information. And for instance, if you have a mutation, you're looking at the sequence, what type of information can you extract from the sequence that could help you predict the effect? For instance, let's consider we are trying to identify mutations that are pathogenic. Okay? What type of information from the sequence you could extract? Any ideas? If you have to develop a method, you're trying to understand pathogenicity, for instance, mutations that will completely abolish protein function, and you're looking at sequence information, what could you use? Any ideas? For instance, you could use evolutionary information, right? So proteins evolve to have a very specific sequence and structure. If you change something that is extremely important for function, it's likely to affect function. And one way to access evolutionary information is via conservation. And you can have also structure-based approaches. And the type of information we can have from protein structures goes beyond evolutionary information, but actually physical-chemical properties that have to do with the actual function of the protein. So structures tell more than the sequence. So let's consider the case that we want to predict pathogenic mutations from sequence. So given a missense mutation X in a given protein Y, we'll have something like this. So usually missense mutations can be given as a code with three parts. So we have a wild-type residue code. Here in blue, in this case, is an arginine in a specific protein position. In this case, it's in position 248. And it's being changed to a mutant, which is a W, which is a tryptophan. So you have a mutation, arginine to tryptophan in position 248. And this happening on a specific protein, let's say TP53, which is a tumor suppressor protein. Okay? So the question is this likely to affect protein function? So changing this residue in this position in this protein, is it likely to affect protein function? And one of the things we could use to try to address that question is calculating conservation. How do we calculate a residue conservation? That's a good exam question. Maybe I asked it. Yeah, anyone? How do you calculate conservation? The idea behind conservation is the extent to which a residue is conserved is an indication of its likely essential role in viability of protein structure and function. So the more conserved a residue is across a family of related proteins, the more likely that a mutation on that residue is going to alter or demolish protein function. So mutation on conserved residues are likely to be pathogenic, or more likely than the rest. So how do we calculate conservation? Anyone? Grace? How do you calculate conservation? I think we mentioned this before. So we've definitely covered the A-specific methods that could give you conservation. Almost there. Almost. It has to do with trees. But it has to do what you actually generate to calculate a tree. What's the first step when you're trying to develop or create a phylogenetic tree? What's your input for? Yes, a multiple sequence alignment. So from a multiple sequence alignment, what you're trying to find is in a family of proteins, so the same protein, so a homolog in different species, whether that specific position is conserved or not. And if it is, and you mutate it, it's likely that's going to be pathogenic. So how do we calculate sequence conservation? It starts with multiple sequence alignments. So it will be a multiple sequence alignments of orthologous sequences. What are orthologs? Is it clear what's a homolog? What's a homolog? What's a homolog? Protein with the same ancestor. What's an ortholog? So differences between orthologs and paralogs. It's been a while, I know. But. So paralogs are from a duplication event. Orthologs are from a speciation events. Basically, if you say two sequences are orthologs, they're from different species. Okay? So basically on the alignment, you want, let's say, myoglobin, which is a protein responsible for storing oxygen, right? In our muscles, for instance. And it binds iron and everything. So you want to identify orthologs, which are myoglobins in different organisms. For instance, you can't see from here, but it's bovine, human, mouse, rat, etc. And then you align them and ask the question, is this position conserved or not? So, but there are considerations and limitations to using this information, for instance, to predict pathogenicity. One is, how do we choose orthologs? Of course, we need to use a criteria, and of course, our conservation calculation will be relative to that criteria. And also the number of orthologs. If you think about this region here, you have a really good coverage here. You have a lot of sequences for which each of the residues positioned. You have several species there, right? So you have a lot of coverage. You have a lot of evidence to say, well, I believe this residue here in orange is very conserved. Can you tell the same about this one? So this region only have 3 or 4, so you don't have enough evidence to say, well, all of them are, let's say, leucine, so it's 100% conserved. The fact is you just don't have enough information. So the number of orthologs in each position of your multiple sequence alignment is a limitation as well. And also, conservation calculations do not distinguish between overall rare and common mutations in a population. So since I'm going to selecting one ortholog, which is basically one sequence per species, I don't have any representation of population variation. So this, I think the second one here is a human protein. This is just a reference. Is it representative of every single different populations? Not really. So that's one of the main limitations of sequence conservation. Okay? And these, of course, will affect and in a certain way limit the methods based on conservation. And the first method based on conservation I'd like to discuss is called SIFT, and it's probably one of the most widely used. So SIFT is sorting intolerant from tolerant. And it's a very, very widely used method for identifying damaging mutations, was one of the first ones. And it's based on a very simple premise, that is, conserved residues are likely to be pathogenic if you change them. So the basic idea behind SIFT is it's identifying homology. Okay? And the workflow or the algorithm for SIFT is very, very simple. It works as follows. So given an input sequence, okay, you have an input sequence and a subset of mutations you like to investigate their effects. You're going to search for related sequences. So you have your input sequence. You ask the question, considering, for instance, a sequence database, what are my orthologs? And I'm hoping by now you know, given a sequence, what type of method you're going to use to ask the question, what are the orthologs on this database? Given that you recover a set of orthologs, then what you do a multiple sequence alignment. And from these multiple sequence alignments, what you have is homologous positions, residues that are homologous. And what you need to do for SIFT is basically calculate a conservation score and a substitution probability. Based on what you see, what's the substitution probability from one residue to the other. And based on that probability, you're going to say, if this probability is over 0.05, well, that mutation is actually tolerated. If it's less than that, the mutation is deleterious. So it's a very, very simple example. Limitations of SIFT. If you don't have enough coverage on your multiple sequence alignment, of course, you won't have a very high confidence prediction. But the method will tell you, I think this is probably pathogenic, but I'm not confident enough because you don't have enough evidence. Okay? And an interesting thing about SIF, as long as you have enough sequence information, you can calculate this for any organism. Okay? An alternative to SIFT, using a few other interesting things, is polyphen-2. Polyphen-2 is basically polymorphism phenotyping version 2. It's also a widely used tool to identify pathogenic mutations in humans, and it's almost a gold standard. When someone, for instance, arrives in a physician appointment, they suspect they have a genetic disorder, they sequence the exome. First thing they're going to do is, given a subset of mutations, run them through polyphen to ask whether any of them are likely to be pathogenic. So it's a widely used tool, and it's a hybrid method. So it still considers homology, but also considers the structural features when available. So more than just asking, is this conserved, it's likely to appear, is actually asking questions, for instance, where the mutation appears, whether the mutation appears in known active or binding sites, which are important for function. Therefore, you would consider them to be pathogenic, whether are in different proteins in different regions, for instance, in transmembrane regions. And also asking the question, how is this mutation affecting protein physical chemistry? Okay? For instance, how this mutation is affecting hydrophobicity, protein interactions, and so on and so forth. So basically, given an input, what polyphen does is calculates a series of sequence-based features as well as structure-based features, and use these to train a probabilistic classifier based on a naive Bayes algorithm that we'll cover on week 12. However, this is only made for human proteins, but it's the gold standard when you're trying to predict pathogenicity in humans. So SIFT and polyphen will give different scores. And they do have different scoring systems. For instance, SIFT will say given mutation is either tolerated or deleterious. If the probability is less than 0.005, it's going to say it's deleterious. If it's more than that, tolerated, while polyphen uses the opposite. So values closer to 1, it will say this is probably damaging, but it also classifies mutations as possibly damaging, benign, and unknown based on whether they have enough information to make a prediction or not. Okay? And the main current limitations of methods that predict pathogenicity is that they overestimate the number of pathogenic mutations. So if you recall from variant calling, so different variant calling methods, they need to find a compromise between two properties, sensitivity and specificity. The main problem with these methods is that this has been shown for population variations, they overestimating one of the classes, right? So saying everything is pathogenic, make sure you're very sensitive, but it's not very specific. So the main challenge now is developing new methods that are more specific. So we do need new, better methods to understand pathogenicity. And this is common to all methods, not only these two. So most of the methods tend to overestimate pathogenic variations, to the point that when people started doing large-scale population variation analysis, for instance, on the NOMAD dataset, they discovered that on average, if you go through polyphen or SIFT, run every single mutation we have, compared to a reference genome, you're likely to find 50 mutations that are predicted to pathogenic, that are predicted as pathogenic per person. We would all be dead. So this is clearly wrong. It's a very serious overestimation, and it's a very serious limitation. So please do, if you work with this, develop new methods to try to better understand pathogenicity. That's extremely important. And an alternative to using sequence conservation is a new concept. This was proposed in 2017, which is tolerance. Instead of asking the question, is this position conserved across evolution, is actually looking at the population and asking the question whether this position accepts missense mutations or not. And this is called the mutation tolerance ratio, or MTR. And it's basically looking at the proportion of missense variants in relation to the proportion of synonymous variants. Okay? So for both observed and expected variation under neutrality, considering synonymous and non-synonymous have the same likelihood of appearing. And this has been done with NOMAD and with a few other exome resources, accounting for over 240,000 exomes, 240,000 different people. And what they do is they calculate what is called this MTR score, which is basically then the proportion of missense variants observed over the total missense plus synonymous, and all of that considering also the potential ones or the expected ones. And with that, you have a ratio from 0 to 1. And what they do is they calculate it for a specific amino acid region instead of per residue base. So they have a 31 codon sliding window, which is basically I consider the current residues, 15 residues before, 15 residues after, and I calculate the MTR score for that region. And I'm basically saying for a 31 amino acid region, what's the missense tolerance ratio of that region. And then you can estimate this ratio per window. And if you plot it alongside the sequence, you see something like this. You see the expected average for a genome, and you clearly see regions that really, really don't tolerate missense mutations at all. So you can ask the question, are there regions, specific regions that are less or more tolerant to missense mutations? And you can also ask the questions, are there genes that are completely intolerant or tolerant to mutations? And this is in the context of a population. So you can use this, for instance, for gene prioritization. And it's been shown that pathogenic mutations are enriched in low MTR regions, so regions that don't accept any missense mutations. And when you look at this, for instance, this is an MTR profile for a cyclin-dependent kinase, CDKL5, and you look at this region, it doesn't really accept many missense mutations. It's probably an indication that it's a functional region. And if you look at this plot, which is basically the domain here, it's a kinase, and you see a lot of pathogenic mutations in that area, saying that it doesn't tolerate. So if you remember, this is calculated with NOMAD and other exome aggregation consortia, which is looking at healthy population. And most of the mutations seen in that area, which doesn't tolerate missense mutations, are actually pathogenic, which makes sense. So they are functionally important regions, and they're important and interesting tool for visualization. So this is called a lollipop plot, that you show the proportion of missense mutations on a given position. And it makes up for an interesting way to visualize where mutations occur. However, all of this is looking at the sequence. And we know that over the past 20, 30 years, we've been accumulating a lot of biological data in terms of sequencing efforts, in terms of sequencing exomes, sequencing tumor samples, and relating those information with clinical and patient data, as well as solving protein structure. So we have a wealth of information that we can leverage on to develop structure-based methods. So we have all the experimental information we need to develop new methods that are more precise, that are more effective than sequence-based ones. So, and the question is now, how do we go and predict effects of mutations on protein structures? So given a protein structure and a specific amino acid residue, so this is the TP53 structure, and this is the arginine 248 position, I want to ask the question, how do we predict the effects, all the potential effects and mechanisms of mutating this residue on the protein structure and function? And amongst the desirable characteristics of such methods are, I want a quantitative prediction. I want to be able to say, if I mutate this arginine to tryptophan, that's more serious than changing it to leucine or other residue, okay? And I want it to be scalable. I want to be able to evaluate that for 1,000 or millions of mutations. And also I would like methods to give me indications of potential molecular mechanisms. For instance, whether this mutation is affecting protein folding or it's affecting protein interactions. And this is very important to understand the roots of pathogenicity, okay? And mainly the relations with phenotype. So you can have a phenotype which underneath in molecular level you have multiple potential molecular mechanisms, and that can help patient treatment, that can help on drug development process. And today I get to talk about what I do, which is cool and not so cool, but I'm going to focus on one specific approach to develop structure-based methods to understand mutation effects on proteins, which is called structural signatures. So what's this structural signature approach? So basically, we understand that several biological entities can be modeled as networks and graphs. So graphs are a very intuitive way, for instance, to model protein structures, mainly because protein structures are a polymer of amino acids that acquire a 3D structure due to atomic interaction. So it's very intuitive to model proteins as interaction networks between amino acids. So amino acids as nodes, interactions as edges. And the question once we model our protein as a graph is how do we capture or model protein shape and chemical composition? Protein shape is important for protein function as well as chemical composition. And the goal then is how do we extract relevant features from this given network to better describe its topology and composition. And what we've been using is the concept of graph-based signatures. So what's a signature? This is a signature that belongs to a very famous person, a very important person. We wouldn't be here if it wasn't for him. Who is this person? Who this belongs to? Alan Turing. What Alan Turing has done. Who is this dude? I don't know. You need to watch Imitation Game. It's a very, very good movie. And it talks about Alan Turing. He's the father of computer science. Every processor is by definition a Turing machine. Okay? How do you know this is Turing's signature? So what your brain does is it identifies a pattern saying this is an A, this is a T, this is a G, and you put everything together and you interpret it. But basically what you did was looking for a pattern. A graph-based signature is a pattern that univocally identifies a graph, in the way that a signature is something that univocally identifies you. So when you look at protein graphs, what we're trying to do is what are the sets of features that univocally identifies, for instance, pathogenic variants, and how we distinguish those from those that are benign. So it's a very simple analogy. But the way we implement this is using a concept called cutoff scanning matrix, or CSM concept. And it's as follows. So we'll get our proteins and model them as undirected labeled graphs. And the information we want to extract to represent its topology is a distance pattern between the nodes. And we'll represent this distance as a cumulative distribution function as follows. For instance, here I have the protein structure of whale myoglobin. And basically for protein structures, what you have, for each atom, you have the X, Y, and Z coordinates for which you can represent as a sphere of different colors. For instance, here we have carbon as gray, oxygen in red, nitrogen in blue. And the way I could generate an undirected label graph from that is basically saying I'm going to select a centroid per amino acid. I'm going to label it based on its physical chemical properties, whether amino acids hydrophobic, positive, negative. And I'm going to say amino acids are interacting if they are within, let's say, 5 angstroms. And this will generate this following graph. And if I count how many labels, I can have one position on a cumulative distribution function. And the cutoff scanning part is basically, if I vary how I generate this graph, I'll have a series of graphs and a cumulative distribution or a probability distribution of the number of labels I have. And if I generate this per label type, let's say between hydrophobic amino acids, between positive and negative, I'll be able to represent both topology and physical chemical properties. So I can use and represent this as vectors, and for each mutation or for each protein we'll have a vector, and collectively for multiple mutations I'll have a matrix, so therefore it's called cutoff scanning matrix. So it's basically varying how I generate my graph and counting how many labels. So a very, very simple and scalable approach. And what we did was use this information that represents topology and physical chemical properties to train supervised learning methods. And what we did first was actually trying to create a mutation analysis platform that came with 3 different flavors. So we used this information together with PRONIT and PROTERM databases, which were experimental databases on protein stability to train a predictor on protein stability, and then use the SCAMP database to train a predictor on protein-protein affinity, and PRONIT on protein nucleic acid affinity. And these were made available as user-friendly web interfaces. And over the years, what we've been doing is adding more flavors to it. So these are basically 3 potential molecular mechanisms by which mutations can affect protein structure and function. And basically what we've done is creating web-based, easy-to-use computation platforms. And we've been adding quite a few of them. So we have today methods to predict effects of mutations on stability and dynamics, on interactions. For instance, this one is new versions of nucleic acids and protein-protein interactions, but also on antibody-antigen interactions. And this is probably the first method capable of predicting effects on protein-drug interactions. And we keep adding more flavors. And these will basically tell us different potential mechanisms of mutations. And today we have around 16 tools and databases that are hit over 1 million times per year from 100 different countries. So it's been widely used. And while these tell us very interesting things and we're having fun developing methods, this is useless as long as is applicable to real-world scenarios. So the question is, yes, we have a very interesting mutation and analysis platform. What can we do with it? So how do we translate all of these methods into real-world applications? How we actually change patient treatment and patients' lives with that? Otherwise, it makes no sense. It's pointless, okay? And I'm going to briefly talk about 2 case studies. One of them is how do we use this information to understand cancer risk prediction, and how do we understand molecular mechanisms in disease, and how important it is to predict. And hopefully I'll manage to convince you that it's extremely important to, more than just saying this mutation is pathogenic, I need to understand by which mechanism. So the first case study, I started doing this when I was in Cambridge. It was on a specific disease type called VHL, or von Hippel-Lindau disease, which is related to kidney cancer. So basically, VHL is a genetic condition. It predisposes individuals to have clear cell renal cell carcinoma, kidney cancer. And we had a cohort of patients with history of VHL, and some of them with high and low risk of developing cancer. And instead of saying, well, if you have a mutation, regardless of what is in VHL, you need to be screened, we need to take care of you because it's likely that you're going to develop kidney cancer. What we did was we used our computational platform to try to segregate mutations in molecular mechanisms, given that we know that VHL interacts with other mutations, for instance, characterizing mutations that are affecting stability and affecting protein-protein affinity, and how this links with clinical experimental data we have. And what we did was, based on that, develop a meta predictor to predict risk of renal cell carcinoma, which went really well. So it achieved that around 95, 98 precision. And what happens with people with VHL in the UK before this was the following. You have history of VHL, you need to be screened for cancer every 3 months or 6 months or something like that, regardless of your background. And the fact is, just the fact that you have a mutation doesn't mean you're going to develop cancer. So what we did is actually develop a method that more precisely will be able to say whether you are in a low-risk group or in a high-risk group. And that can have transformative effects. So we created a database for a hospital in Cambridge called Addenbrooke's Hospital. And that's the part that a computational method that can be translated and actually change how we treat and manage patients. So this changed how patients are screened at the hospital. So instead of just saying we're going to screen you every 6 months, if you are on the high risk, we'll keep doing that and perhaps even do it more often. If you're on the low risk, we don't need to be so worried. And that has 2 effects, 2 very important effects. One, saves money to the public health system, which is NHS in the UK. So they're saving over £10 million for the next 10 years. And this is one protein. And this is a very rare disease. So it's one protein, 1 year of work. Imagine what we can do with so many different rare diseases out there, okay? And this has transformative effects for the patient as well. If you know a patient is on a high-risk group, you're likely to catch cancer as early as possible, increasing risks of treatment success as well. And the second case study, which also started in Cambridge, was in a specific metabolic disease called alkaptonuria. It's a very rare disease, and actually is the first ever described metabolic disease. It was described back in 1902, and It occurs because of mutations in a gene called HGD, homogeneous dioxygenase, and it basically is on the pathways of tyrosine metabolism. And basically, this protein forms this beautiful large homocomplex. So it's 6 chains coming together, form a dimer of trimers, so they interlink between each other, meaning there's a lot of protein-protein interaction going on. And what happens in alkaptonuria is if this protein doesn't work, this compound is going to be accumulating connective tissue, including, for instance, cardiac valves. Prognosis is not very good. So what happened is I was working in my first postdoc, and someone knocked on the door from Develop ACure, a guy called Nick Suho, and he's heterozygous for AKU, and his wife as well. And both his kids have AKU. AKU has absolutely no cure, no drug so far. So he refused to basically let their kids die without a chance. And basically now he goes around the world getting academics to work together to try to develop drugs for alkaptonuria and also raising funds for clinical trials. So what we did there with him was basically trying to understand the mutation effects on alkaptonuria and understanding potential molecular mechanisms of protein malfunction. And at that time, there was a clinical trial going on called SONIA-2. And surprisingly, what we identified using computational methods was that mutation effects correlated with patient response once we stratified patients. Okay? So we were able to classify the molecular mechanisms using computational tools into 3 groups. So mutations affecting the active site of the enzyme, mutations affecting protein folding, and mutations affecting protein interactions. And if we look at this graph here, which is actually the response to treatment on the SONIA-2 clinical trial before and after treatment, the drug was actually only working for people with one of these 3 potential genotypes. So alkaptonuria is one phenotype with 3 different molecular mechanisms. Instead of just saying, well, this has failed, actually was rescued for people with a specific genotype. So this is understanding the molecular mechanism of the disease. So we came up with a lot of hypotheses using the computational tools that were then validated experimentally. And this is a very embarrassing photo that I always show, but they're very important. Why? I'm a computer scientist by training. So what I did there was actually went to the lab and did protein expression, purification, crystallization. I didn't put anything on fire. I did learn quite a few things about biochemistry and how proteins work, how mutations work, and more importantly, we were able to validate our predictions, and we achieved a really high correlation with experimental data. So in a year, we created a new method that helped people segregate patients in a clinical trial, and we could better understand the molecular mechanisms of the disease. And this is important. So understanding the molecular mechanism is even more important than saying if a mutation is pathogenic or not. And the to-go-home message here is the importance of interdisciplinary work and the importance of bioinformatics and computational biology. So if I can give you one, but only one advice is don't be afraid to cross discipline barriers. That can be extremely important. So use as many opportunities as you have. So if you come from a computer background, try to learn from biochemistry, and the opposite as well. If you come from a biological background, try to learn to code, because that combination is extremely powerful. Okay? And that's all for today. Thank you very much.